Hi, everyone. I'm Erin Massey. Today, I'm going to be explaining protein digestion by drawing it on a board. Um, so the first thing I wanted to do is walk everyone through the main parts of the GI tract. And then we'll go from there, looking at each one individually. So a main overview, we have the mouth. Not much digestion of protein specifically. Actually, no digestion of protein is happening in the mouth. Fun fact, in the mouth, in the saliva particularly, you have amylase. ACE letting you know it's an enzyme. Amylase primarily works on carbohydrates. So if you were to eat a cracker and you were to hold that cracker in your mouth, producing more and more saliva, and the amylase would over time work on that cracker. The cracker would grow in sweetness that you were able to pick up on your tongue, on your taste buds. So we're breaking down carbs mostly in the mouth. A little bit of amylase, or a little bit of lipase is produced, but really it's mainly carbs. There is mechanical digestion happening. Chewing, that's what we have these canines for, right? Breaking down protein. So if you were to eat a chicken, you'd be able to mechanically break it down and do something called a bolus. Bolus. A lot of food. That's what that is, right? That bolus is then going to travel down the esophagus. If your upper esophageal sphincter is behaving correctly and you're paying attention and you don't choke or aspirate, it successfully goes into the esophagus. If you aspirate, though, it can literally go down the wrong pipe into the lungs. And that's why we have older people who get weaker upper esophageal sphincter stop working as well. They're more likely to choke because those muscles aren't as strong and they get aspiration pneumonia with literally food going into the lungs. So hopefully everything's working properly and it goes through the esophagus. That bolus travels down. Still no digestion happening, just half the transport. Finally, we're going to get to the stomach, which has one big important enzyme to remember, and we'll review the pathway that gets us there. The small intestine has quite a few enzymes that are working there, 5 in particular we need to remember. And the large intestine, digestion by our enzymes stop, but our digestion of bacteria really amps up. And then the colon, a little fermentation has been known to happen in the colon, but really it's just excretion at that point. So I'm going to pause here and come back and we will talk about the stomach. And we're back to talk about the stomach. Not the best drawing, but okay. Now the stomach, the bolus that is now this wad of food that's coming down is actually going to activate a specific hormone called, so gastrin is going to be released. So after going through the lower esophageal sphincter, which I like to mention the sphincters because they're kind of like gatekeepers. This bolus is going to tell the G cells to release gastrin. Yep. Gastrin, cascade with gastrin, is then going to release something called hydrochloric acid. Remember back in chemistry, you never want to touch hydrochloric acid. And this hydrochloric acid is coming from something called parietal cells. The hydrochloric acid is very important because once released, it's going to help emulsify the food that is then entering the stomach. So actually literally break apart that bolus into bigger parts. And the more you chew, the more surface area, the better and faster that hydrochloric acid is able to digest and break apart those molecules. So hydrochloric acid being released. Hydrochloric acid is also going to do something else. Stay tuned. And pepsinogen is going to be released by the chief cells. Pepsinogen is something we call a zymogen. Now, zymogens are important because there's quite a few along this pathway, along protein digestion, that are important to know. A zymogen means that this enzyme is not active. It's present, but not working to do its job yet, which is break down our proteins in different ways. So pepsinogen needs something. It actually needs hydrochloric acid. So that hydrochloric acid is going to let the pepsinogen self-activate and turn into pepsin. So this is the one enzyme we really need to know. Well, a big one. This is the one that's actually going to be breaking down, the worker is going to be breaking down our proteins in the stomach. Specifically, you have a peptide chain. Pepsin likes to work on the hydrophobic, the ones that don't like water, hydrophobic amino acids, the ones that prefer to not react with water, are hydrophobic amino acids. And it likes to cut them at the carboxyl end, where the carbon is. So digestion's released, G cells, hydrochloric acid is then stimulated to release the parietal cells, break apart your food, emulsify your food more, as well as activate pepsinogen into pepsin, which then will break apart long peptide bonds at the hydrophobic amino acids, specifically on the carboxyl end. What next? Well, after going to the stomach, there is a pyloric sphincter. And the pyloric sphincter is now going to let no longer a bolus, this acidic chyme, enter into our small intestine. And while it is small, it does a lot. Stay tuned. So we've come from the stomach, we're journeying into the small intestine. It's important to know the small intestine has 3 parts. Well, most of the action for digestion is going to start in the first part, but continues. As far as parts of the small intestine are concerned, you have the duodenum, the ileum, and the jejunum. That's backwards, you guys. Duodenum, jejunum, ileum, I comes last. If you have any great mnemonics for memorizing that order, please share them. I think that's those are important to know. So duodenum, again, a lot of our action is going to happen in the duodenum and small intestine. So when this acidic chyme enters the intestines, a hormone is going to be released, the boss, called secretin. Secretin is secreted. I think there's an I there. Yep, there's an I there. All right, so secretin is secreted. Doing a couple things. The first is going to release bicarbonate. So bicarbonate is going to be released, and bicarbonate, this chyme's really acidic at this point. Well, the intestine would really like that pH to go up a little bit so those enzymes can work properly. So this bicarbonate is going to calm the acid down a little bit, increase the pH, so it then can be worked on. And at the same time, this is not accurate, but for to scale, but the pancreas, which is hanging out here somewhere, looks like kind of a corn cobby, is going to pump its juices into the first part of the small intestine, the duodenum. That's why mainly things happen. It stirs the duodenum. All those juices are their strongest there. And there are 5 of those to know. All zymogens, all inactive at that point, are being pumped into the small intestines. We'll go back to those. So at the same time, secretin is released, bicarb's happening, things are getting pumped, other things are happening at the same time. The first one, something called enteropeptidase. Is released to help break the proteins apart, as well as CCK. CCK is actually responsible for enteropeptidase, but CCK does some other things too, which are very important in the cascade from the enzymes that are coming from the pancreas. So the first enzyme that's important to know is trypsinogen. Trypsinogen is important, very important to know, because CCK is going to activate it into trypsin. Which then starts a whole bunch of things. Before we move on to the other things that are going to happen, I want to stop and pause here and say that what does trypsin do? Like, why is it important anyway? Well, it does a whole bunch of things. In of itself, trypsin likes to break down peptide bonds, just like pepsin. Specifically, the amino acids that happen to be basic. So it likes breaking apart basic amino acids at the carboxyl end, just like pepsin. And it's important to know it is an endopeptidase. So it's going to break apart those basic amino acids somewhere in the middle of a peptide bond, not on the ends. So likes basic amino acids. What else does trypsin do? I said it was important, right? Well, I said there were 5 of these enzymes in the to know in the small intestine. Trypsinogen is one of them when not activated. After trypsin has been activated, it can activate the other 4. So it is responsible not only for breaking apart peptide bonds, but activating the other 4 big enzymes we're going to learn about. So the other one is chymotrypsinogen. With the help of trypsin, becomes chymotrypsin. And it likes to break apart amino acids that are aromatic. Again, at the carboxyl end. All of them are going to be at the carboxyl end. They really like cutting into that C, that carbon. Number 3 is proelastase, which with trypsin, again, becomes elastase. And this really likes the, I'm going to say aliphatic, but I'm not sure that's how you pronounce it. Aliphatic amino acids. The ones that look like fatty acids, the branch chain, as well as glycine and alanine, kind of look like fatty acids. Small chains, small group chains. So They both like aromatic, and this is important to know. Keep this in mind. You can't see it on the screen. Sorry. They both like aromatic. One likes aromatic, one likes the ones that kind of look like fatty acids. It's important to know. Now there's two more. I'm going to change colors. Trypsin is still in charge of activating them from zymogens into their active form. And they have very similar names. Pro-carboxy peptidase A and pro-carboxy peptidase B. So why did I do these in a different color? So they're a little bit different in how they work. They still use trypsin to be activated, and they become carboxy peptidase. So you get rid of the pro A and carboxy peptidase B. They still like to cleave amino acids off of the and break apart peptide bonds. What makes them special is that they're actually going to get the chance to make even smaller molecules and just single amino acids. So while the other three are producing smaller peptide bonds, these two get to produce amino acids, specifically amino acids that are aromatic and aliphatic. Why? Because they're cleaving off the end. So they're just going to cut one amino acid off of the end that happen to be aromatic. So they're really taking the products of proelastase and procarboxypeptidase. They're taking the products of chymotrypsin and elastase. So procarboxypeptidase A and B are taking the products from the two above it and making them into smaller proteins. From here, we have small peptide bonds that have been broken down by these enzymes, as well as individual amino acids. Now, some of the amino acids and dipeptides and tripeptides can be taken in by the cell through transport-mediated absorption, usually a sodium or hydrogen potassium pump that puts it into the cell. But some of them might still be too big. So things like tetrapeptides, so if there's more than 4 amino acids, that tetrapeptide can't be absorbed yet. So it's going to need to be worked on by something called aminopeptidase, which is found in the cytosol on the brush border of a cell. So aminopeptidase will help further cut that down. So things are being absorbed, but we don't always absorb 100% of our protein, right? So what happens to that protein that we don't actually digest? Well, it travels on to the large intestine. So in our large intestine, we have a couple things to help those undigested and unabsorbed proteins, called bacteria. This is a heated topic right now in nutrition. People refer to it as microbiota, gut bacteria, gut health, really popular anywhere on TikTok, right? Gut bacteria is really important. The gut makes up 80% of our immune system is one of the statistics you'll find out there because of the bacteria that live there. But a big amount of our bacteria are living right in our large intestine. So one of the big things they're responsible for is fermenting fermenting those leftover proteins that don't get digestion. They'll help absorb them for us by breaking them down through fermentation. But fermentation, it's a smelly process, right? No, it really is. You're gonna produce things like CO2, short chain fatty acids, and even some ammonia, byproduct of the bacteria and fermentation. Some proteins in particular cause odorants that are particularly memorable to me. Putrescine. If I can spell it right. Putrescine and cadaverine. What makes these memorable to me and makes them even grosser, if a smell is putrid, it's really, really bad, like rotten eggs, right? And cadaverine reminds me of dead bodies, like a cadaver. It's really dark. But those things are being produced in the large intestines, all thanks to our bacteria that keep us healthy. Other things are absorbed in those intestines, though, that I did not mention, which probably should have mentioned first. There's three big things that are in our intestines that are absorbed in the intestine. The biggest one is water. So, as things go through the stomach and we need water to help hydrolyze our proteins, water is actually going to be absorbed here in our intestines. And that's what helps us make the output, right, our feces. Minerals are absorbed here, as well as volatiles, toxic compounds that can then be filtered by the liver, and excreted in urine. So water, minerals, volatiles, very important, happen in the large intestine. Another gem to know is that those bacteria, they don't just ferment, but that fermentation process can also produce, as well as there's vitamin K. So a lot of our vitamin K can be synthesized in that large intestine. And what happens after the large intestine? The colon, removing things for excretion. I think that about does it. If you have any questions for me, be sure to put them in the comment section, like and subscribe. No, let me know if you have a great mnemonic for the small intestine and the parts of it for memory. Thank you.